For this next video clip for Chapter 12, we're going to talk about some various antimicrobial proteins, so protein molecules whose job it is to help fight off these invading microorganisms right away after they enter the body. And there are several different examples of these. First one we're going to talk about are some proteins called interferons. Now, there are various types of interferon. We mentioned those in an earlier video clip, and the ones that we're mainly talking about here are interferon alpha and beta. There's an interferon called interferon gamma that is a big time signaling molecule between various cell types in the immune system, and it has lots of other jobs. But more specifically here, we're going to be talking about interferons alpha and beta and their job in helping limit the spread of viruses inside your body. So this is kind of interesting how all of this works. So over here on the left-hand side, you've got a poor little cell in your body, and it has been infected by a virus. So the virus has entered, it released its nucleic acid, it's multiplying, it's assembling new viruses. We talked about all of that back in Chapter 5. Now, very often, your cells have methods for detecting that a virus is present. One of them is the presence of double-stranded RNA. So that's not a common feature of RNA inside human cells, but it is associated with RNA viruses. You know, RNA viruses have to make complementary copies of their RNA genomes when they're replicating. So one of the signals can be the presence of double-stranded RNA in your cells. That's a signal to one of your cells that, hey, there's an RNA virus in here, so we better set off the alarm bells and warn our neighbors. So when that happens, interferons are proteins. So you have a gene on your DNA that codes for these, or genes, and when viruses enter your cells, that gene gets activated, it gets expressed, so it undergoes transcription and translation to make these interferons. They get released from this virus-infected cell, and basically this is a warning. It's a warning signal to neighboring cells that a virus is present and get ready to take on the enemy. So these interferons spread over to neighboring cells. They bind to receptors. So there are kind of like hormone receptors on the surface of the cell to which these attach. And when that happens, it signals this neighboring cell over here to activate genes, start expressing genes that code for antiviral proteins. So these are proteins, they're primarily enzymes that are going to limit the ability of many different viruses to replicate inside the cell. So if one of these viruses over here that came from this cell manages to infect this cell, the replication will be limited or even stopped. So you might wonder, well, why don't our cells just make these antiviral proteins all the time? They sound like great little shields that they could be using. And the problem is, one of the ways that these antiviral proteins works is it shuts down protein synthesis inside this cell. So yes, that's going to stop the virus, but you can't have that happening all the time, because then the cell would not ever be able to make its own proteins and it would die. But if you do that temporarily, it can help limit the ability of an invading virus to replicate inside that host cell. Also, some of the antiviral proteins degrade RNA of viruses, and that's another way that they help limit the replication. All right, the next topic is going to be a little bit more in depth, and it's about a special protein-based system that's part of your innate immunity, and it's called the complement system. And the reason it's called the complement system, this is like if one thing complements another, they fit together well, they help each other out. So it's that kind of use of the term complement. And so this is a system of proteins, and it benefits both the innate and then also the adaptive immune system, the third line of defense that we'll learn about when we get into Chapter 13. So I've got some diagrams and so forth from a previous textbook that we used here, because I think they do a little bit better job of explaining this complement system than your textbook does. You know, different textbooks have different pros and cons associated with them. So I'm going to give you a little bit more information on complement to make it more understandable, I think, than your textbook actually does. So again, the complement system is protein-based, so we're not talking about cells here. And there are actually over 30 different kinds of complement proteins. They're made in the liver. And if you've had Biology 202, this might be reminding you of clotting factors. Most of your clotting factors are proteins that are made by the liver. And it's kind of the same story. So when you guys study blood coagulation in Biology 202, you learn about how your clotting factors are produced by the liver, and they're always circulating around in your blood in an inactive state. And then they basically get clipped, and that activates them and allows them to play their role in blood coagulation. And it's kind of this, it's the same thing with the complement proteins. They're always circulating around in your blood in an inactive form. But when they get activated, they work very, very, very rapidly. You've got like this rapid cascade of events that happens to help fight infections. This is part of your nonspecific immunity. It's not adaptable in any way. So, these complement proteins are always available. You don't start making them in response to infection by a particular microbe. The three ways that these complement proteins help destroy microbes is by cytolysis, so cell lysis. So it can make, especially bacterial cells, burst, but other types of invading cells as well. Triggers inflammation. So complement proteins will, function kind of like the chemical signals released by mast cells and basophils. It will help stimulate inflammation. And then also enhancing phagocytosis. So remember how we've talked about a lot of your pathogens have ways of getting around phagocytosis early in infection. The complement system is going to be one of these things that counteracts that and helps get these microorganisms phagocytosed anyway. And as I was mentioning, your complement proteins are inactive in the blood, so they're in a bigger form. For example, C3 is an inactive complement protein. These complement proteins are mostly named with a C designation for complement. And then they get clipped to activate them. It's like C3 would get split into two smaller proteins, one called C3A and the other one called C3B. And these two are active forms. Okay, so how does this work? So again, there are a whole bunch of different complement proteins, but and there are three different major ways that this complement cascade gets activated, and we'll talk a little bit about those. But they all kind of home in on this one particular type of complement protein called C3. So this is in your blood, and it can get into your body fluids as well, and this is an inactive form. In order for it to become activated, it has to get cut or split into two smaller proteins, one called C3A and the other one called C3B. So this is kind of the critical complement protein here for getting this system to work. All right, and then what does C3B do? So C3 is non-functional. You got to have it clipped in order to do anything. And then C3B does a couple of nice things for us once it gets made. C3B can attach to many different microorganisms. It'll attach to their membranes. And then C3B is like a magnet for phagocytic cells like neutrophils and macrophages. So again, remember we talked about how pathogens have things like capsules on them, or when we talked about the M proteins of Streptococcus pyogenes in Chapter 11, things like that that help them avoid phagocytosis. But if a microbe gets coated with these C3B proteins, those phagocytic cells have receptors on their surface that attach to C3B, and that's going to help them achieve phagocytosis. This coating process is called opsonization. We'll hear that again in Chapter 13 when we talk about antibodies. So these microbes have become opsonized with C3B. All right, then another thing that can happen is C3B can also function as an enzyme. So it is capable of clipping or cleaving another complement protein called C5. So this is an inactive complement protein in your blood and in your body fluids. When C3B is made, it clips this into C5B and C5A. C5B, once it's available, it sets off a chain reaction. So it attracts in some other complement proteins called C6 and C7 and C8 and C9. and they assemble together and stick themselves into the membranes of microbes and create what are called membrane attack complexes. And basically you're forming a channel. You can see a pore forms, and that causes the poor little microbial cell to burst. So that's a membrane attack complex. But see, this all starts up here. You got to get this C3 clipped first, because if you don't clip C3, you won't have C3b. So you won't have any opsonization over here. And if you don't have C3b, C5 is never going to get clipped. If you don't have C5b, you won't be able to bring in the C7, C6, C8, and C9 to assemble together these membrane attack complexes that are going to poke holes in the membranes of microbial cells and cause those cells to lyse. So it's all about getting that C3 up here activated. Also notice here on the diagram what happens to the other half of C3a and the other half of C5. What happens to the other half, C3a, and the other half is C5, your C5a. Those are both two little proteins that are good at enhancing inflammation. So they will stimulate mast cells, remember you've got those in your connective tissues, to dump out all of their chemical signaling molecules like histamines and so forth. And as we've discussed, that will stimulate inflammation. So this complement system is kind of nice because it will, in 3 different ways, help you fight off infections by enhancing phagocytosis, by causing these pores or membrane complexes to poke holes in the membranes of microorganisms, and then also by increasing or stimulating inflammation. All right, but how do we get this to happen? This is critical. You got to have these C3 complement proteins. They have to get split into C3b and C3a. So there are actually 3 different ways that can happen. Now this slide actually is basically just summarizing what I've already discussed on the previous one, but you've got it here in a text format if you want to take a look at that. Oh, one thing I didn't mention here, though, gram-positive bacteria are more resistant to these membrane attack complexes that we took a look at over here. Can you guess why? What do gram-positive bacteria have that might be a barrier to these membrane attack complexes? And if you're thinking back to chapter 3, remember your gram-positive bacteria have a very thick peptidoglycan cell wall, so the cell membrane is actually deep to that thick cell wall, so these membrane attack complexes can't assemble as easily on their cell membranes because the complement proteins can't get through that big thick peptidoglycan cell wall layer. Okay, and again, how do the complement proteins enhance inflammation? Remember, it's these 2 guys over here, C3a and C5a. When they get made, they help enhance inflammation, and they do that by attaching to mast cells. Mast cells have receptors for C5a and C3a, and when one or both of those attach, the mast cells dump out all of their chemical signaling molecules that promote inflammation. C5a is also can function in chemotaxis. So neutrophils and macrophages are attracted to it. Remember, chemotaxis is like a chemical trail of breadcrumbs. So if you've got an infection going on and complement has been activated, you'll have these C5a proteins around, and those are like magnets for neutrophils and macrophages to come swarming in and help you fight off infection. So kind of a neat little system here, this complement system that, again, it's assisting the other things that we've already talked about in helping you fight off infection. All right, so again, with complement, the key is to get this to happen. If this doesn't happen, all of these other nice things are not going to occur. So how do we get that to happen? There are 3 ways that have been discovered so far. One is called the classical pathway. One's called the alternative pathway. And there's a third one, I think that's the most recently discovered, that's called the lectin pathway. So we'll take a quick look at what those are. The classical pathway involves antibodies. Antibodies are Y-shaped protein molecules that are, now those are produced in response to very specific molecules or markers on the surfaces of microbes that are invading the body. And so we'll learn about antibodies when we get into chapter 13. So just keep this in mind. One of the things that antibodies can do is activate this complement cascade that helps kill off microorganisms. So antibodies are weapons made by your third line of defense that will attach to the surfaces of microbes. When that happens, there's another complement protein called C1 that's attracted to antibodies. So if you have antibodies attached to a microbe, then C1 can come in and dock onto the kind of the stem portions here of the antibodies. And then once this happens, you've got two more complement proteins, C2 and C4, that get involved. So C2 and C4 come in, and C1 clips C2 into C2A and C2B, and it clips C4 into C4A and C4B. So it's one of these, like, it's kind of like a chain reaction type thing, kind of just very similar to what you guys learn about when you study blood coagulation in Biology 202, where one step leads to the next and leads to the next and leads to the next. Notice the C2A and the C4B get together and functions as an enzyme that clips C3 into C3B and C3A. And that's what we needed to have happen for all of those nice little things to occur, our opsonization, our cell lysis, and stimulation of inflammation. So this is one way that you can get this to happen. So you need antibodies attaching to the microorganism here to attract in the C1 to set off these other events. All right, that's called the classical pathway of complement activation. Then there's an alternative pathway. So there are some special complement factors called B, D, and P. And these are complement proteins that your body makes, and they have a strong attraction for certain lipid carbohydrate complexes on the surfaces of microorganisms. Okay, so when they attach, they then bring in C3, and the formation of all of this together helps clip C3 into C3b and C3a, and then that leads to all those nice things that we've already talked about. So you don't need those antibodies here. These are proteins that just have a natural, strong attraction for lipid carbohydrate complexes that you would find on the surfaces of microorganisms. And finally, there's a third way that has been discovered for activating this complement system, and this involves molecules that are called lectins. So lectins are proteins. They're made in the liver, and they're strongly attracted to carbohydrates. So many microorganisms are going to have some carbohydrates decorating their surface molecules. So these lectins are very good at attaching to carbohydrates that you would find, carbohydrate patterns, sugar patterns that you would find on the surfaces of many microbes. So these are actually going to be a type of PAMP. Remember, PAMPs are pathogen-associated molecular patterns. So little 3D shapes of pathogen-associated molecules that you would not find on the surfaces of our human cells. They're microbial molecular patterns. So lectins can attach to carbohydrate patterns that are associated with microbes. And then once that happens, that triggers the activation of C2 and C4, like we saw with antibodies. That causes C2A and C4B to get together. That functions as an enzyme that clips C3 into C3B and C3A. And now you get all those nice, fun little things that the complement proteins help out with. So, kind of a nice little protein-based system that has 3 different ways that we know about of getting activated, and then plays a really important role in enhancing the fighting of infections. You may down the road hear about, so there are different types of immunodeficiencies that patients can have where part of their immune system just isn't functioning. And so there are patients out there who have complement defects, and they particularly have problems with fighting off bacterial infections. So the complement system is really, really important for that. Your complement cascade must be regulated very, very, very carefully because it is very powerful. You don't want membrane attack complexes accidentally assembling in the surfaces of your own cell membrane. So our cells actually have inhibitory proteins that are sitting in the membranes of our cells that prevent those membrane attack complexes from forming. The levels of complement proteins are kept fairly low, so you don't accidentally trigger it and have these complement events damaging your own cells. And then once the complement proteins get activated, we actually have enzymes that quickly digest them and get rid of them so that you don't have an ongoing complement response that could potentially damage your own cells. And as I mentioned, there are some people who have genetic defects that cause them not to produce one or more complement proteins, and they do have. Now, they can still fight off infections, but they're probably going to be a lot slower at being able to accomplish that. All right, so now complement virulence factors. Remember the virulence factors we learned about in Chapter 11. Pathogens have virulence factors, and so some of the pathogens do have ways of getting around the complement system. And remember those capsules that are present on the surfaces of many pathogenic bacteria? Those can actually inhibit the complement system from getting activated. And those M proteins on Streptococcus pyogenes can actually also play a role in inhibiting activation of complement as well. So we've got these nice weapons. They always have these pathogens have these ugly weapons that can fight back. All right, last but not least, now we mentioned this a little bit earlier, but just as a reminder, there are other antimicrobial peptides or proteins that non-specifically help us fight off microorganisms. Most of these are about 20 to 50 amino acids in length, so they're fairly short. That's fairly short for a chain of amino acids. And what these do is they are able to attach to the membranes of microorganisms, and they create pores. So we already talked a little bit about defensins and dermacidins when we were discussing barriers. There's another type called protegrins. So you have these produced and available at your barriers, and they're pretty good at poking holes in the membranes of microorganisms. This is what the diagram from your textbook looks like. So these peptides or proteins, they have a positively charged portion, and that is attracted to your phospholipids. The phospho part of your phospholipids tends to have a negative charge, so that brings those, helps bring those two together. This diagram I like a little bit better because it's actually showing a defensin and how its 3D shape, once it inserts itself into a membrane, actually helps create a pore. So you've got those around as well, and there are researchers who are trying to figure out, can we produce these artificially, commercially, and maybe use them as a new type of drug? Yes, we do produce them naturally, but what if we actually gave them artificially to patients and in higher amounts? Would that potentially be like a new type of antibiotic drug that we could use to help fight infections? All right, so that's actually going to conclude Chapter 12 on the innate immune system. Now we're finally going to move on to the third line of defense as we move into Chapter 13, and we're going to talk about specific immunity and also the very important topic of immunizations or vaccinations. That'll be the final thing that we'll talk about in Chapter 13.